My lab is interested in trying to determine how genes that are expressed in specific cells in the brain regulate the activity of those cells and allow them to mediate their function in the regulation of behavior. So our work intersects with the clinical side of psychiatry by working with human geneticists such as Evan Eichler and Ray Fournier, who actually are involved in identifying genetic mutations in patients that have neurodevelopmental disorders such as autism. So working in collaboration with those individuals, we can identify the genes that are dysregulated or mutated in human patients and then study the function of those genes in a translational model system. In order to accomplish this, we've developed a modified version of the CRISPR-Cas9 methodology. And essentially CRISPR-Cas9 is a bacterial immunity defense mechanism that bacteria have evolved over billions of years to inactivate viral genomes during infection. So using this technology, we've developed actually a viral-based delivery system for this CRISPR-Cas9. So we can deliver using a viral gene delivery method the CRISPR-Cas9 to target specific genes in any cell type of the brain of the mouse that we're interested in investigating. Since we developed this method, we've shared it with numerous labs around the world as well as numerous labs here at UW, and we find that it's a highly efficient and highly reproducible means of studying gene function in neural circuits of a variety of different species.
In my lab, our main program follows a genetics-first approach to studying autism. The reason is autism is an incredibly heterogeneous disorder. We know there's a large number of different genetic variants that contribute to autism. And we know that because we've been involved in a lot of gene discovery projects here in the lab. What we identified is that there's hundreds of different genes or genetic variants that are contributing to autism. And so we want to understand, well, how do these different genetically defined subgroups, how do they look different from other individuals with autism, and how do they look similar? So what we do is we identify these particular genes of interest. We work with our collaborators in neuroscience to understand, well, what does that gene do? What is the, what happens when you disrupt that gene, either through functional models or animal models, what have you. And then we say, well, we can take that information about what might be different and then figure out what assays, what methodologies can we use to identify what those disruptions do to humans. So for example, we've identified individuals who have disruptive mutations to a particular gene called CHD8. And so what we do is we recruit everyone that we can find who has a disruptive mutation to that gene, and we then look at how does the brain function by using EEG. We look at how does the body develop. So we collect information with 3D MD imaging to generate images of the face. We can perform calculated measurements of those individuals. We use eye tracking to understand how are those individuals attending to their world. What we've done at the Autism Center is we've made sure that every single person that walks the door has the opportunity to participate in a research project. So we've built into the process for check-in, it's built in right onto the medical record screen that shows up, ask this family about participating in research. So families can tack on to their appointment with their provider a research study visit. This is particularly important for our low-income families who can't afford to take time off work, can't afford to actually even get into the center, and need to utilize some sort of Medicaid-supported transportation service. So we can now invite these families and truly have the family participate in a research project where historically they have not participated. So that's the biggest way I'd say we incorporate diversity in inclusive practices into our research here. I mean, it really does change the way autism research is done.
My study, the Brighton Study, was funded by the National Institute of Mental Health, and the purpose of the study was to see whether or not we could do a randomized clinical trial completely on people's smartphones. We were originally thinking it would take a while to get 150 people to sign up for the project, but what we actually found that after 1 week of deploying ads through Facebook, Craigslist, we had recruited 150 people within a couple of days. In 5 weeks, we were able to screen 3,000 people and enroll 1,200 people into the study. That was the first wave, which was done completely in English. So in addition to being able to recruit a really representative sample of the US into this project, we were also able to collect data in a way that you can't do in traditional face-to-face trials. So people, through their phone, downloaded an assessment app that not only collected daily ratings of mood that we captured like in the moment, but also captured information about how physically active they were based on the accelerometer and GPS on people's phones, how socially engaged they were through numbers of text messages, phone calls, and emails sent and responded to. NIMH has been very interested in this project as well and has used this study as an example in presentations about collecting large-scale data, and that this is something that should be encouraged. And also, we need to think about how to come up with strategies to keep people engaged in these studies longer. The Brighton study touches on the theme of diversity and inclusion, and that we're able to pretty much break down a lot of barriers that keep underserved populations from coming to research centers. And a lot of it is, they don't have the time to come in, the assessments are burdensome. This allows them to participate at a pace that they want to participate in. They can do it from the freedom of their home, they can do it privately, they don't actually have to come see me or come to university. It also allows us to recruit people who aren't near an academic center where research trial is being run. So that's another way that it addresses disparities, I think, in research.
Well, thanks to funding from the department for a pilot study that I've been working on, I am really interested in thinking about the aspects of place where people go throughout their activities and thinking about how those might be related to marijuana use and craving in young adults. So to do this study, we are linking two different types of research methodologies. One is ecological momentary assessment. So the idea of EMA is to really try to figure out how to measure people's behaviors and mood and other feelings, perhaps, in their natural context. So the way we typically are doing this, or how people in the department are also doing this, is to prompt people to answer short surveys multiple times a day for, say, in our case, a month. And then we will be trying to link that to where they're going, and we'll be doing that using GPS. Down the road, there are some opportunities, I think, that it could exist for integrating GPS and EMA. So for example, I think there is opportunity or potential to create interventions that can target individuals at certain high-risk locations or at high-risk moments. Say, for example, that we do find that, the greater exposure to marijuana retail outlets could trigger cravings or could trigger someone to actually use marijuana. Then we could use the GPS embedded within someone's smartphone to do what we call geofencing. And so when someone gets near, say, a marijuana retail outlet, we could give them some sort of prevention message. In terms of trying to link GPS and aspects of where people are and marijuana use, I think this is one of the first, if not the first.
Accountable care organizations are transforming how healthcare systems function. Traditionally, they focus on maximizing the effectiveness of treatments delivered to patients who present for care, and now they are responsible for managing the health of an entire enrolled population. So the methods for evaluating interventions in accountable care organizations are very different. Traditionally, we've looked at the effectiveness of an intervention for patients who get it, and now we're interested in population-level effectiveness, which depends on the clinical effectiveness of the intervention, how many patients it reaches. Reach depends on access for patients and the capacity of providers to deliver the intervention. To address this problem, we're working with healthcare systems to develop the infrastructure to collect patient-reported outcome data from patients outside the context of a clinical encounter. Not only does this allow us healthcare systems to monitor and manage the population health of an entire enrolled patients, but it also allows us to have complete data on patient-reported outcomes, including data from patients who are not engaged in care. So this allows us to do pragmatic trials more efficiently and effectively.
In John Fortney's PCORI-funded pragmatic trial, we've had a nice opportunity to develop a mobile platform that includes a patient smartphone app where patients can send in their symptom scores like the PHQ-9 for depression and send those to their care provider, who then sees them in an existing web-based patient registry like the one we have here. This allows the providers to do a better job adjusting treatments for patients who are not improving. Now we're trying to learn more about what it takes to incorporate these sorts of tools into routine clinical workflows. When you do something like this well, it's really a win for patients who can then update their providers at their own time more conveniently and also track along with their own progress on their own. They also have the opportunity to receive care in a more timely fashion, so this is really patient-centered. It's also a win for providers because providers don't have to administer and then document the symptom scales themselves, and so they save time. And it's a win for health services researchers who can obtain more complete outcome data than would be possible at traditional clinic visits, and also get data on how patients are doing more frequently than you would get in a typical clinic setting or with research assessments that may be many months apart. Tools like this that allow healthcare to become more accessible to diverse communities can really help engage patients who have not been well served by traditional healthcare settings and also haven't been well represented in clinical research.